Next presentation will be given by Özlem Türeci. She's the co-founder and current CEO of Ganymed Pharmaceuticals in Mainz. She's involved in many different activities in the Mainz arena, co-initiator of the cluster, individualized immune intervention, and many other things. And Özlem will give us an overview about biomarker-supported development of an immune-eector-mobilizing antibody.
Yes, thank you very much, Dirk, for this introduction. As Dirk has already pointed out, the subject of my talk is one of the immune therapy drug development programs we are pursuing in what we call the Mainz Immunotherapy Hub, which is a private-public partnership which we have created over the last years in order to bring innovations from the academic space, innovations as such you are all involved in, viably to the patient's bedside. And the specific project I would like to introduce today, IMAP362, a monoclonal antibody against a novel target which we are developing in solid cancers, is a project which is pursued by one of our biotech companies, Ganymed, and our non-profit translational institute, TRON. Monoclonal antibodies are a well-established treatment modality in cancer therapy. They are around for nearly 20 years now, clinically and also commercially successful. However, there is space for improvement, and this space is actually the target level, which is targeted by the monoclonal antibodies. For many years, the field has basically restricted itself in assessing growth factor receptors and their ligands as targets for monoclonal antibodies. And in the field, in the arena of non-solid cancers, which is of non-hematological cancers, which is our field of interest, this meant that the target space is quite restricted. In the first decade of antibody development, antibodies against only four targets have reached the market and the patient's bedside. Only recently, a second target space basically opened, which are the checkpoint inhibitors, and we all are very excited about this. And basically, what has happened here is that a novel target discovery space of a multitude of potential target candidates has opened up. What we are pursuing in our projects is a third complementary path to this. We are interested in targets which don't have primarily a role in the function of the tumor cell or the immune system. We are more interested in targets which are cancer cell specific, targets which are not expressed in any normal tissue type at all, or if they are expressed there, are not accessible to IV administered antibodies. So this is an entirely different concept. And the antibodies we are interested in developing are antibodies which are not primarily blocking any function, but are designed such that they bring potent killing mechanisms to the tumor. There are a number of such technologies which have been matured over the last decade: bispecific antibodies, ADCs, toxin conjugates, CARs, for example. And for these sorts of therapies, or immune effector recruiting antibodies, we are searching for targets. The project I want to introduce to you today is such an antibody which adheres to this concept, IMAP362. Its target is Claudin 18.2, a novel cancer cell-specific surface molecule. Claudins are major components of tight junctions, and as such, this Claudin 18.2, has the typical class-specific effects, namely that it is involved in the sealing of intercellular sheets and in paracellular transport, polarization of epithelial cells. In addition to this, we have shown by siRNA technology that this target also has a tumor biological role. It is involved in tumor cell proliferation, in chemokinesis and chemotaxis, and is associated to ERK and PKC signaling pathways. But what was important when we selected this target for antibody development was its cancer cell selective expression, namely the fact that it is not expressed at all in the majority of normal tissues, which are all listed here, neither on the transcript or on the protein level. The only tissue in which Claudin 18.2 is expressed is actually stomach mucosa, which means that here we have a highly selective lineage marker, in this case for stomach. We know this paradigm already from non-solid, from hematological cancers, where we have such highly selective differentiation markers, and we use them for clinically meaningful therapies. CD20, for example, is an example. In the solid cancer field, they are rare and do not exist so far. Stomach tissue expressing, does that mean that we have to fear toxicity? We have shown preclinically and in the meantime also clinically that this is not the case. And the reason for this is simply that we have a tight junction molecule here, which in the healthy epithelia of normal cells is protected from IV and administered antibodies. However, with breakdown of tight junctions and loss of cell polarity being one of the first hallmarks of cancer, in cancer cells, this target is accessible. It's not so trivial, I have to admit. You have to find the right epitope in this molecule, which is cryptic and hidden in such a normal architecture of epithelia, but it is doable. We have shown this, that this concept of differential accessibility is viable in relevant animal models, in mice and cynomolgus, which express the same epitope, which is bound by our antibody. In all those studies, we have never observed toxicity, neither systemically nor specifically in the gastric tissue. This is an example for one of these studies where we have shown that IV administered IMAP362 ends up in a human gastric cancer xenograft expressing the target, but nowhere else, even if we try to detect this antibody IV administered in the primary tissue of the treated mouse. In terms of expression in cancers, Claudin 18.2 is quite broadly expressed. It is expressed in gastric cancers, which is not a surprise, as it is a lineage marker for this tissue type, so that along malignant transformation, Claudin 18.2 expression seems to be sustained. However, and surprisingly, also cancer types, cancers of esophagus, pancreas, lung, ovary, and biliary duct, which in the normal tissue counterpart do not express this target, activate Claudin 18.2 upon malignant transformation aberrantly, which means it's basically switched on. So that in summary, we here have a target which is pan-cancer expressed in basically a number of high medical desperate prognosis indications, and which has a very high selectivity with lack of expression in many normal important tissues, which means that basically we could combine this with different killing mechanisms. And we actually have done this. We have shown preclinically that ADCs, antibody drug conjugates, for example, if you conjugate auristatin to IMAP362, do cure mouse tumors in syngeneic models. We have shown that using this target as a target for bispecifics does the job in mouse models. So in principle, we have animal proof of concept for different types of equipping such an antibody with killing mechanisms. In terms of clinical development, however, we have decided to go for a naked antibody which was engineered to induce ADCC and CDC, so that this is the mode of action, the dominant mode of action of IMAP362. And the reason for this was basically that as immunologists, we had the vision that inducing inflammation at the tumor site could help us to alter the tumor microenvironment to our favor, and might at the end even end up in some sort of auto-vaccination. So this was the vision. So these are the dominant mechanisms of IMAP362. In addition to this, we see some apoptosis induction upon cross-linking and direct proliferation inhibition. In oncology, where everything is about a combination treatment, the question is certainly how do these mechanisms translate if you combine with chemotherapies. And here there are basically two key messages I would like to share with you. One is that we found out that treating human cancer cells, gastric and pancreatic cancer cells, with the chemotherapeutics you wouldn't use in these tumors clinically results in upregulation of the target in these cells and in a higher density of surface epitope expression for our antibody. And this directly translates to a shift of the dose response curves for ADCC and CDC to the left and upwards, which basically means that chemotherapy pre-sensitizes human cancer cells for modes of actions of this antibody. A second observation we have made is that in cancers, in animal models, where we treat mice with a combination of this antibody and especially immunogenic cell death inducing chemotherapeutics, the EOX regimen, for example, epirubicin, oxaliplatin, capecitabine is such a regimen which is composed of immunogenic cell death inducers. We get heavy infiltration of these combination treated tumors with T cells. This is shown here in an immunocompetent mouse experiment where mice were inoculated with a claudin 18.2 expressing mouse tumor and were treated either with chemotherapy alone, with IMAP alone, or a combination of both in an induction maintenance scheme. And what you are seeing here basically are residual tumors at day 17, which only in the combination treatment group show total eradication of any claudin 18.2 positive tumor. So all the remaining tumor cells are either immune escape variants or immune escape variants. What we also see in parallel is that these tumors show an infiltration with heavy infiltration with T cells, and this translates to a sustained survival benefit of these mice, so that we seem to have an introduced an immunological difference here. What certainly is of major interest for us is, do we have adaptive T cell immunity? We have some indications for this from the mouse models, and this is certainly a hint we are further pursuing, because this could mean that such an approach could be a next-gen immune oncology approach where you not only unleash immune suppression from prevalent T cell specificities, but can create new ones. So clinical translation. The indication we have chosen for this is advanced gastroesophageal cancer, and the program we are conducting here is directed towards getting a proof of concept for the first-line treatment of gastroesophageal cancer, IMAP362 in combination with a standard of care chemotherapy regimen in claudin 18.2 positive cancers, which are HER2 negative. So this would be the the indication. 4 clinical trials are either completed or still ongoing. 3 of them shown here are early trials, phase 1, phase 2A, in which we have used IMAP362 in the setting of pre-treated patients as a single agent treatment or in combination with an exploratory immune modulatory protocol as a salvage treatment. In addition, we have an ongoing phase 2 randomized trial, 250 patients treated in 3 arms, in which IMAP362 is tested in the very indication we are interested in, namely first line in combination with chemotherapy. In combination with this therapeutic drug, this one is what I wanted to show. First, in combination with this drug development, we are developing a companion diagnostic type, IVD, because it appears obvious that target expression levels of Claudin 18.2 in the patient's tumors could have an impact on the response we would get with such an antibody. We have not shown this so far in the clinical setting, but we need a tool in order to investigate this in a diligent and viable way. And for this, we have developed immunohistochemistry IVD kit, which allows us to detect and quantify Claudin 18.2 with this fully analytically validated CE marked kit. And this kit is basically concurrently developed and assessed in terms of its clinical utility in the clinical trials we are performing with IMAP362. Let me first summarize briefly the data we have from the first 3 trials I mentioned, which is single use in advanced patients, at least second line, most of them later, and in progression upon recruitment. In these trials, in terms of safety, we have basically proven our hypothesis. We have shown that the antibody is very safe, well tolerated. We don't see any DLT, dose-limiting toxicity, even at the highest doses. And the only related relevant adverse effect basically is vomiting and nausea, transient and mostly G1, grade 1 and grade 2. In terms of efficacy, as I said, the antibody in this trial was tested in the single agent setting. We prefer, because we expect activity there, the chemotherapy combination, which is part of the ongoing trial. However, as single agent, we have observed some activity in our so-called mono trial, which administers the antibody repeatedly. We have seen 4 partial remissions of later than second line treatment patients and 8 stable diseases of patients who were entered into the trial under progression. In terms of clinical pharmacology, these trials helped us to determine our recommended phase 2 dose, which is in the range of 300 to 600 mg per square meter at least. So the now ongoing trial is for us the so to say, clinical proof of concept trial, the so-called FAST trial, which is right now ongoing in multinationally at more than 50 clinical centers in 5 countries. And this trial uses the CLAW-Detect kit, which is our IVD kit, in order to screen patients prior recruitment. We have decided to go for an enrichment type of trial, which means that only patients who have a high expression level, at least 2+ intensity membrane staining in at least 40% of their cells, are entered in this trial in order to increase the probability of success. I personally believe that we can give this antibody to patients with any expression This is, however, something one has to tackle and explore in a clinical trial, and this we would do in a phase 3 trial, a trial who would be open for lower expressors as well, and need then to bring the data to show which expression threshold is the one which means profiting from this trial. Patients are randomized into 3 arms. First arm is the maximum 8 cycles of EOX chemotherapy, which is the control arm and one of the standard treatments. Arm 2 and arm 3 combine this with IMAP 362 in 2 different doses given 3 weekly, and as IMAP is so well tolerated, the treatment is pursued as maintenance treatment until progression. So this is the typical first-line population. Key primary endpoints is progression-free survival and safety, and key secondary endpoints is overall survival and certainly also clinical utility of the IVDD as predictive marker. We have an extensive biomarker program which is directed towards understanding the immunological mechanism of action of this antibody and whether it complies with the vision with which we started, which means that we look for SNPs of important immune effector mechanism related molecules, chemokines, Fc receptors, complements, etc. Moreover, in selected long-term survivors, we are looking for adaptive T-cell immunity, primarily with regard to antigen spreading to mutated epitopes in the very patient's tumors, so that this will hopefully help us to understand the mode of action of this antibody in the clinical setting. What we already can say is that also in combination with chemotherapy, we see safety and well tolerability of the antibody. There's not much added on the backbone toxicity of the chemotherapy we see. In terms of efficacy, we will see more within the next month because the final analysis of this data is being expected within the next months and will happen in January next year. So is this a concept which is one time and only feasible because there are not more targets fitting to this paradigm? In fact, we think that if this concept shows viable, this would open us an additional novel target space, a third one. We have a second target which also is in clinical development, which basically follows the same blueprint as described for Claudin 18.2 and IMAP 362. And this target, Claudin 6, is even a bit more interesting because it is highly tumor selective. As an embryonic marker, it is not expressed at all in any normal tissue, also not on the transcript level, but is at the same time a cancer stem cell marker, which means to our knowledge that this is the first cancer cell-specific cancer stem cell marker. So to summarize my talk, what we have found is that there are also in solid cancers targets of high cancer cell selectivity. We have shown that you can generate ADCC and CDC inducing antibodies against them. Whether this is a viable clinical context, the answer to this question is basically imminent. We will analyze the data soon, and what I can already tell you is that we will have a very nice story to tell, and I hope that we can see you at the ASCO in summer next year in order to bring the continuation to this story. Thank you very much.
Thanks a lot, Özlem. Maybe I can start with the questions. In patients that ultimately progress under treatment, do you still see the antigen expressed, or is there variation of antigen expression level in progressing patients?
Yes, unfortunately, we could not do such sorts of studies taking post-treatment biopsies from stomach cancer patients. So that at the moment I cannot answer that. I hope that we can follow this up with investigator-initiated studies where we can look more closely really into the clinics of single patients in order to understand exactly this.
Günther.
Very interesting approach. So I wonder about it, you addressed it briefly, the accessibility of the claudin proteins by antibodies. Because in intact tight junctions, they should not be well accessible. But are they accessible if you try to do staining on, let's say, live junctions in a three-dimensional structure? Or does your antibody mainly react with parts of the tumor where the junctions break up? Then the antibody, the antigen is accessible, and then somehow a chain reaction starts, and more junctions are being broken up.
Yes, this is exactly how we think it works, because if you look into sections of also human tumors, you see that there are cells which have lost their polarity, but there are also cells which still are polarized, and you see tight junction type of structures. So in many well-differentiated tumors, you have a mixture of both. But on the other side, you also have tumors which are entirely de-differentiated, also in gastric cancers, where you don't see really any even abortive tight junction formation.
And if you look at tumor spheres in vitro,
we have not
which I think they form tight junctions.
Yes.
Does the antibody then stain these?
We have not looked into spheres, I have to admit.
Okay.
There's many data on expression levels of claudin 18.2 or 6 in, let's call them tumor-initiating cells.
Cancer stem cells, yeah. For claudin 6, we have shown that in cancer stem cells we see claudin 6, but also in their sort of followers, not only in the cancer stem cells. It is obviously maintained over different cell types also after the cancer stem cell. For claudin 18.2, we have no proof for this so far.
Okay. Any other tumor types that are interesting for claudin 18.2 antibody approaches?
Yes, pancreatic cancer is for sure very interesting. We see even higher and more homogeneous expression in patient specimens for pancreatic cancer, even though in that case it's de novo switched on, which is quite amazing. Ovarian cancer, 80% of the mucinous ovarian cancers with a very low prognosis do express claudin 18.2. Lung cancer, around 6%, which does not sound much, but actually would be a huge segment of patients who could profit if this works, and over 80% of biliary duct cancers do express. So basically all high medical need cancer types.
Last question. Looking at the expression pattern, is that a target that would be suitable for a CAR-based approach, or is it not? Is the expression not really specific enough?
We think it is. Preclinically, we have supporting data for this, also for other high potent approaches like bispecific T-cell engagers, for example, so that this tight junction concept seems to be really tight in the normal tissue, and we don't see any toxicity in animal models.
Okay. I don't see any other questions, so we have I guess a 30-minute break. Thanks a lot.